In this video, we're going to look at the proteins that are responsible for recombination. And we're going to start with Rec-A. We know Rec-A is important for recombination because studies with E. coli that don't have Rec-A have shown that they have recombination rates are roughly 10,000 times lower than normal. And we have actually talked about Rec-A before. It is a protein that stimulates Lex-A for the SOS response in E. coli. Now what Rec-A does is that it will bind to either single-stranded or double-stranded DNA that has a single-stranded gap. And it's highly cooperative. So we talked about cooperativity with hemoglobin, but what that means is when one Rec-A binds, other Rec-As want to bind even stronger. So we bind to DNA that has our gap, and then we're going to bind to homologous double-stranded DNA. Because remember, this is recombination, so we need homologous DNA for this. Then we're going to catalyze our strand exchange using ATP. And for assembly to even begin, you need about 5 to 6 copies of your Rec-A. And here we can see what it looks like. So we have 1, 2, 3, 4, 5 different copies of Rec-A, and our DNA is wrapping around this Rec-A. And each Rec-A, as shown in this image, has extensive contacts with its neighbors. So we have a lot of interaction with the DNA going around it. And what happens is that this DNA is connecting through the sugar and phosphate backbone, which makes a lot of sense. You don't know what DNA will get a single-stranded nick in it, so you don't want to be looking for specific bases. You're going to be wanting to just bind to the phosphates of DNA. Now let's look at the overall process for doing strand exchange. So first step is that ATP in single-stranded DNA will bind to our Rec-A complex, and that will be our initiation complex. Then we need double-strand DNA to bind the initiation complex, and we're going to form a 3-stranded helix from that. After we have our 3-stranded helix, Rec-A is going to rotate the bases of the homologous strand to basically do a strand exchange, where now blue and red are DNA. And during this process, since we are moving molecules and that takes energy, ATP will be hydrolyzed for this. Now, to make sure that only the complementary DNA is being exchanged, our single-stranded or blue DNA is going to be kept in the normal beta conformation. And when DNA is in the B conformation, it can really only accept normal Watson-Crick base pairs. So this is how we're making sure we are exchanging our complementary strands here. Now, Rec-A is in prokaryotes. In eukaryotes, the protein is called Rad51. It's homologous to Rec-A, and from the research that has been done so far, Rad51 seems to be doing the same job as Rec-A. Now here's another image of what I just talked about. Each purple molecule is Rec-A. So right now it's bound to our single-stranded DNA, which is green. Here we have red double-stranded DNA coming in, and here's our triple helix forming, where we have 2 strands of red DNA and 1 strand of green. But spooling out, we have made a green and red DNA, and single-stranded red DNA is coming off. So that is how Rec-A works by zooming out a little bit more. All right, so let's look at the other players who are part of this process now. So Rec-BCD. So Rec-BCD is going to help create the single-stranded DNA for Rec-A to bind. So BCD are our SOS genes. So BCD are only turned on after about 50 minutes or so of DNA replication being halted, or it's once the SOS genes. That might be shorter than 50 minutes, but DNA replication's halted. We can't get past certain block. Turn on the SOS device to try to get past this blockage, and recombination is one way to do it. So B, C, and D have both a helicase and a nuclease, and what they'll do is they'll bind double-stranded DNA, and then they'll start chopping away at that DNA. They'll unwind it through the helicase and start degrading it through the nuclease. Now, the 3' end, which is blue, is chopped way more frequently than the 5' end, which is red. So you can see blue has smaller pieces than red. And this will continue until on the 3' end we hit the chi sequence, which is GCT, GGT, GG. And this sequence, the chi sequence, is encountered roughly every 5 kilobase pairs in E. coli. So we're not going to chop too far until we hit the chi sequence. Once we hit this sequence, the 3' end will stop chopping, but we're going to continue chopping away the 5' end, and on the 3' end, Rec A will also bind. So Rec B is now going to recruit to Rec A, and now we are ready for the Rec A to help with strand insertion, like strand invasion, like we just talked about on the previous slides. Now let's look at what this BCD structure looks like, the enzyme. And B and C, so B is yellow, C is this teal color, they're interwound. They're associated a lot. The C terminus domain of B right here is our nuclease, and it's connected through this tether, this 21 amino acid tether that connects it to the body. Right? And so what happens, the way this works, is that we have 15 base pairs of double-stranded DNA entering through this hole between Rec B and Rec C. And there's a part of Rec C here colored in green that forms a wedge to break apart the DNA, to make it single-stranded. Now the 3' end is closer to the nuclease domain than the 5' end. So that is why the 3' end it gets chopped up more often, because it has easier access to this nuclease domain. The 5' end has to compete, but it's in a less favorable position, so it gets chopped less often. However, once we bind that chi sequence, the 3' end can no longer go to the nuclease domain. That's why we see an uptick in the amount that 5' is cleaved. Now, we're going to turn back to holiday junctions at this point and look at the proteins that are involved for holiday junctions. So we need RUV A and B for holiday junctions branch migration. So RUV A, that's a homotetramer, so that's what we're looking at right here. It looks like a flower, and right now we are bound to two different double-stranded DNA. So one strand was initially green and yellow, and the other strand was teal and purple. The red's oxygens, and the orange here are phosphates, and the blue are nitrogens. So this flower, we have 4 different grooves, and that allows us to bind the 4 different arms of the holiday junction. In the center, we have 4 negative charge projections, or pins, and this is kind of directing the DNA to split, or that's the idea, because the DNA's phosphate bonds are negatively charged, so when we come across one of these pins, we don't want to be near it, so we're going to split off. And that is how it is thought that RUV A is helping to take in our double-stranded DNA and make new helixes out of them. Now, RUV A is not the only player. We have RUV B. So here is an idea of how we think RovB works based on micrograph images like this. So RovB forms a hexamer on each side of RovA. And what happens is that they're bound to DNA, and they kind of pull the DNA into the grooves of RevA. So they're the force that's kind of pulling the DNA towards RevA. And because of these pins, the DNA is being pushed in, and then they split, and they are forming new DNA here. Now, how do we resolve this holiday junction? Well, that's a new enzyme called RevC, which is our nuclease. So the idea with how we cut this apart is that one of these tetramers will have to dissociate. And when it dissociates, RevC kind of takes its place, and there's still another copy of RevA still there. So the RevC will take its place, make the cut like we talked about in the last video, where we looked at the more cartoon image of the holiday junction, and seal up the nick with ligase. And remember, it's either going to cut at the strands that were exchanged or not exchanged, and based on if they do that, you'll get DNA that looks different. But the proteins that are responsible for that are RevA, B, C, and DNA ligase, and this is the image of what it actually looks like. And with that, that ends this video. I will see you in the next video.